So good afternoon, guys. Today I'm going to teach about the protein, introduction to protein, and different types of protein structure, and what are the different types of bonds involved in the protein, and different types of configuration and conformation in the protein, and how to determine the protein structure and properties of the protein, and different things which we are going to discuss. Last session, I think we have discussed about the amino acids. So before moving to the session, I will give the connectivity between amino acid to the protein. So basically, amino acid, I think if the linkage of amino acid, one amino acid to another amino acid, in the same way, the linkage will be useful to form a protein. So that's we have started initially with the amino acids. I think in the last session we have covered the classification of amino acids, amino acid classification based on the structure, based on the nutrition, based on the different things which we have discussed, and I have shared the complete amino acid notes with you. So in that, all types of classifications are there, and along with that, I have shared one more PDF also. In that PDF, the amino acid classification based on the behavior, like polar amino acid, non-polar amino acids, charged amino acid, and non-charged amino acids. So please go through it, and if any doubts, I think you can ask at the end of the 10-minute session. Today, I'm going to talk about the proteins. So let's talk about the introduction of the protein, then types of different structures of the protein, then bonds involvement and stabilization criteria, then how to determine the protein structure, then properties, then if any time will permits, then we will discuss more. Let's talk about the introduction to the protein. So that's all. Yeah. So the term protein is derived from the Greek word. It's not a Latin word. So the Greek word is a proteus. That's a kind of introduction. As I mentioned, that proteins are the polymers of L-alpha amino acids. The term polymers means that it consists of a group of molecule. In terms of chemistry, when we talk about the polymers definition will change from the one segment to another segment. So in a chemistry prospect, polymers means it consists of a bulk, or it's consistent of a bulk or group of a molecule. Let's say a group of amino acids. So here, which type of amino acid? Majorly L-alpha amino acids. Okay, alpha L amino acid. I think you know what is the alpha, beta, and gamma, delta. L means the configuration. So again, I'm repeating just for the revising the last session. So proteins are the polymers of L-alpha amino acids. So coming to the Belgius is a Sweden chemist, suggested the name of the protein, the group of organic compounds that are at most important to the life. When I say important to the life, means amino acids are the proteins are the very, very important organic a molecule. Later we found that it's a very, very important to the human body. Let's say bio. That's why bio plus molecules. So these are biomolecules. Let's say proteins. Amino acids are these building blocks to the proteins. So these proteins and amino acids are the very important to the life. So this Sweden chemist, by the importance of these molecules, he identified that importance. It is important to the life. Okay, so this is a Sweden scientist suggested that the name of the proteins to the group of organic compounds that are the utmost important to the life. Then everything is going to the picture. So there is importance of the protein. Then it will convert to the receptor. Then drug design. These drug and what are the drugs which we are going to read in the pharmacology? These drug will bind to the certain receptors. So receptors also called as a protein. So there is a important in the life in terms of pharmacology, in terms of energy, like proteins are the very energy richest molecule in our life. So that is the Sweden scientist in ancient days, he told about it. Proteins are the very, very important to the life in terms of energy, in terms of various kinds of mechanisms. A drug will come and bind to some receptor. So these kind of mechanisms, these kind of things which we are reading because of the protein. When we are calling protein, when we are calling receptor, I hope you have a clear-cut idea about it. So I'm not going into that. Then Muller is a Dutch chemist. So he uses the term protein. He used the term protein. How you have used the term? based on the molecular weight. So again, a molecular weight is differentiating between the protein to peptide, peptide to the polypeptide. So this Mulder is a scientist, used the term protein for the high molecular weight nitrogen rich. There are the lot of elements present in the protein structure, basically amino acid structure, but nitrogen is the very highest amount of the part will be involved in the protein structure. I think you have a clear idea about it. So what are the major elements present in the protein structure? Major elements are a carbon, oxygen, nitrogen, hydrogen, sulfur. Out of this, nitrogen is a very rich amount of the fragment or the segment will be involved in the protein structure. So that's why Mulder is a scientist. He told that proteins are the organic molecule containing the nitrogen rich part to the based on the elements which we see, that carbon, oxygen, nitrogen, hydrogen, sulfur. Out of these five elements, Mulder is say that nitrogen is the very richest portion in a protein structure. I hope that is the introduction about the protein. I think again I'm revising that. There are the two ways. Two scientists, they told about the protein. One thing, that nitrogen is a very rich source in the protein structure. Second, that Bergelius told that proteins are the very very important organic molecule to the life. So that is a clear cut introduction about the protein. Second, that the content of nitrogen, I'm going to the fifth point. The content of the nitrogen is an essential component of the protein, as told by the Muller. An average of 60% estimation of the net the laboratory. So when I so here the content of nitrogen, an essential component of the protein, is told by the Mulder. So an average 60% when we are trying to do experiments in the laboratory. So an average, there is a branch, or there is a separate field is called peptide chemistry. By using these kind of branch of the subject, we used to synthesize the proteins, because in our human body we have a lot of proteins also called as receptors. So if you want to design a drug to the receptor to the protein, we need to have a proper structure of the protein. So for that we need to synthesize the protein. I think you have synthesized the molecules or the ligands or the drugs on it in our laboratory. But if you will go to the higher education level, like a bioinformatics or the chemoinformatics, you need to synthesize the proteins also. Once if you have synthesized the protein, then you need to identify the structure. Then we need to determine the structure. Once if we have such kind of things, then we will try to see the interaction of the ligand to the protein. That is called protein ligand interaction. So while doing this kind of experiments, while doing this kind of things in a laboratory, so by using the method, Jaldal method, that is the method name. In the laboratory, the scientists have identified the 16% of the nitrogen is a rich amount of the nitrogen, means the 60%, that is the value of that percentage, is identified the in a laboratory while doing by using this method. By using this method in a laboratory, 16% of the nitrogen can be available in the protein structure. So that is the fifth point. So I hope you understand why the nitrogen is a very richest part in the protein. The Mulder is the scientist which is told by that. Then it is a 16% which was identified in the laboratory using the method, Jaldal method. So that is the kind of brief introduction about the protein. Yeah, I hope if you have any doubts you can ask. Otherwise I can give you a brief again, revise to the again I will revise the first slide. So first slide, I talk about the protein word. It is a Greek word. Then all proteins are the polymers of L alpha amino acids. Then two scientists told about the some things related to the protein in accidents, like before I think 150 to 200 years back, they told proteins are the very very important to the life. Proteins are the organic molecule, very important to the life. Then Mulder is told that nitrogen element is the very richest source in the protein structure. While doing experiments in the laboratory, they have identified the 60% of the nitrogen is involved in the protein structure. They found out by the Jaldal method. So that is the kind of introduction. I'm moving to the next slide. So last In last class, I think we have seen the basic structure of the amino acid. So amino acid structure, if you see the amino acid structure, there will be a primary carbon which will be attached to the single hydrogen, then R group. R group is that kind of specific group will be differentiating from one amino acid to another amino acid. So there is a two default fragments are there. One is that amino group, another one is the carboxyl group. So this is the complete skeleton of the amino acids. So now we are connecting this amino acid to the protein structure. As I told that proteins are the polymer of L-alpha amino acids. When I say the bunch of or the bulk of the group of amino acids will be forming the protein. So this is the basic structure of the amino acid. So now I will give the differentiation between the protein, polypeptide, and peptide. So to form a polypeptide or the protein, I will differentiate how we can differentiate the polypeptide and protein and peptide in the next points. Let's talk about the first point. To form a polypeptide or the proteins, amino acids are joined by the peptide bonds. I will explain what is a peptide bond right now only. So let's talk about what is the peptide bond. Here I told that amino acids are joined by the peptide bond. If we want to make a protein, if you want to prepare a protein, if you want to see the protein structure in a different ways, there will be a strong peptide bond is a formation is required. That is called a peptide bond. Amino acids are joined by the peptide bond. So let's say, as you know the amino acid structure, this is the one amino acid. I think if you see the structure, this is the one amino acid. This is another amino acid. So amino acids are joined by the peptide bond. How bond will form? Let's see. So in which amino acid amino or NH2 of the one amino acid bonds to the carboxylic acid, also called as COH, to the another amino acids. So if you see here, one amino acid of the carboxylic group, you can see C double bond O will link to the another amino acid of the amino bond. This is called NH. So let's say in exam, GPAT exam, if they will ask what is the peptide bond. Peptide bond, I think this slide will be easy to you to understand. Yeah, you can see here a difference between amine and amide. I think you know what is amine. That let's call about amide. Is a organic compound that contains a carbonyl group bonded with the nitrogen atom. So this is called complete group of amide. So same in an example, if they ask what is peptide bond. Peptide bond is a bond linked to the carboxylic group of carbon to the amide amine group of nitrogen. So that is a clear-cut differentiation between amine and amide and peptide bond. So let's talk about, I hope you understand what is the peptide bond. Peptide bond is the bond will form between two amino acids. Means that amino acid will join through the bond. That bond is called peptide bond. So that is a different ways to understand the peptide bond. I hope you understand that. If you want to synthesize the protein, or if you want to synthesize the polypeptide, if you want to see the protein structure, there are the couple of bonds are there. You can see what are the bonds involved in the protein or the polypeptide structure. Let's talk about only peptide bond. I hope you understand what is a peptide bond. Peptide bond is the bond used to link the two amino acids. That's it. Next, now I will differentiate the peptide and polypeptide and protein. Peptide is a 2 or more. More means up to 50. If you see the 50 amino acid link, that we is to call as a peptide. Peptide is a 2 or more. More means up to 50 amino acid joined by the peptide bonds. That is called peptide. So I hope that if the number of amino acid or the linkage of amino acid is 2 or up to 50, that is called peptides. Before forming to the proteins, peptides are the very important. First, we need to identify the peptide, then polypeptide, then protein. Direct protein, it is a very tough job to getting a protein. So whenever we are trying to do synthesis of the protein, first we need to do amino acid synthesis or amino acid kind of reactant, like a reactant A and reactant B. I think you know, and with the formation of the catalyst, then we used to get a product. In the same way, we used to, whenever we are doing a protein synthesis, reactants are the amino acids. So at least two reactants are the two amino acids are the more than up to 50. If you have such kind of the segment, then if you combine with both, then we will get a peptide. After getting a peptide, then we need to identify the polypeptide, then polypeptide to the protein. So that is the clear-cut definition. So this is the one amino acid, this is another amino acid. So I hope that you know what is the basic skeleton. Carbon attached to the specific group R and attached to the single carbon, there is a amine group, there is a carboxyl group, C double bond OH. So amino acid of carboxylic group of OH will link to the another amino acid of H. After while doing this reaction or this condensation, as water molecule will be disassociate, then remaining NH of one amino acid will bond to the CO of another amino acid will be losing with one water molecule will form a bond that is called peptide bond. So clear cut, this is the complete reaction. So by losing the one water molecule, again forming with bond between CO to NH. Now peptide bond means that a bond between C double bond O to NH means that carbon of carbon to the amide of nitrogen. So that is that about the peptide formation. With the peptide bond, either two amino acid or more than two, means up to 50. Next, polypeptide is a chain of many amino acids. When I told polypeptide means chain of many amino acids means chain of peptides means that it should be more than 50 amino acid. If it is less than 50, we are calling as a peptide. If amino acid linkage will be more than 50, then we are calling as a polypeptide. The term is a differentiating. Polypeptide means more than number of the peptides. Means that monopeptide, dipeptide, tripeptide. Example of the tripeptide is the glutathione. I think in BPPK, pharmacokinetics, and I think that subject I don't know that either you covered or not. BPPK in that subject, I think if you go through that metabolism, in the metabolism there is a phase 1, phase 2, phase 1, phase 2, phase 3 metabolism is there. Phase 3 metabolism is a kind of conjugated related metabolism. In that, I think you come across with the glutathione is a kind of glutathione is a one kind of molecule which will be used to undergo one one form to another form. I think you know what is the difference between active form, inactive form, metabolism. Metabolism is the criteria where drug will enter into the body. It will convert one form to another form by using various kind of enzymes or the molecules or the organic compounds. Best one is the glutathione. Glutathione will be useful to do the conjugated metabolism. So example, glutathione is a tripeptide. I think you can write this point. It is a very, very important to the GPAT. Glutathione is a tripeptide. Glutathione, if you see the glutathione structure, it is a combination of three amino acid. Amino acid namely as a cysteine, glutamic acid, and glycine. Guys, please note down this point. Glutathione is a tripeptide combination of three amino acid. I'm giving the differentiation between the peptide to polypeptide. If one peptide, monopeptide, if two amino acid, dipeptide, if three amino acid, tripeptide. If more than like 3-4, then that is called as a polypeptide. Example is the tripeptide is the glutathione. When we say tripeptide, it should be have three amino acids, namely cysteine, glutamic acid, glycine. I think you wrote these points. If not, I will include those points also in slide. I will give later. If not, you can write now itself. We are differentiating between the peptide and polypeptide. When I say peptide, it can be monopeptide, dipeptide, tripeptide. If more than like 4 or 5, then we are calling as a polypeptide. I am giving one example for the tripeptide, which is very, very important for the GPAT, or when we are reading that BPPK, biopharmaceutics and pharmacokinetics. So in that there is a topic metabolism. In that we came to know that what is the different types of metabolism. Conjugated metabolism, the one type of metabolism. In that you will read about the glutathione. At right now you can note down the example for the tripeptide is glutathione. Example for the tripeptide is glutathione. When we say tripeptide means it is a combination of three amino acids, namely glutathione, cysteine, and glutamic acid. Glutathione is a tripeptide. It is a combination of three amino acid, namely cysteine, glutamic acid, glycine. Again, I am repeating. Glutathione is a tripeptide combination of 3 amino acids, namely cysteine, glutamic acid, glycine. Again, I am repeating, glutathione is a combination of 3 amino acids or tripeptide, namely cysteine, glutamic acid, glycine. It is a very, very important. That's why I am repeating again and again. So please note down it and repeat one more time after the session. That might be useful to you to get kind of good concepts in a GPAD. So I hope you understand what is a peptide and polypeptide. Now, proteins contain one or more polypeptides. Therefore, proteins are the long chain amino acids held together by the peptide bonds. Now, if you want to see the protein structure, if you want to synthesize the protein structure, you should have at least one or more polypeptides. Okay, not like a peptide, not like a protein, not like a peptide, not like a polypeptide. If you want to see the protein structure, if you want to synthesize the protein, you should have a one or more polypeptide. That means a huge number of amino acids, means like more than 100. So that's the kind of differentiation between the peptide, polypeptide, and a protein. In this slide, we have seen the basic skeleton of amino acid, then how the linkage between one amino acid to another amino acid, linkage can be formed or linked by the peptide bond. We have seen the what is peptide bond, then we have seen the what is the peptide, what is the polypeptide, what is the protein. Peptide can be monopeptide or dipeptide or tripeptide. Example for the tripeptide, glutathione. Glutathione is a tripeptide combination of 3 amino acids, namely cysteine, glutamic acid, and glycine. So protein is the common, the protein contains one or more polypeptide. So these peptides are the polypeptides linked by the peptide bond. Peptide bond is the bond between the carbonyl carbon to amide nitrogen. Guys, is this clear or should I repeat again one more time? It's a very, very basics and fundamentals. Protein is a very vast topic. I know when we move to higher educations, when we will move to higher education, like BSc Bioinformatics, MTech Bioinformatics, MTech Biotechnology, when we move to that, then you came to know all the things. At right now, I'm only covering for the GPAD portion. These are the very, very basics and fundamentals to the GPAD up to the any competitive exam. If you have any doubt, again, I am ready to repeat one more time. Yes, let's move to next slides. Now we are going to read, or we are going to talk about the types of protein structure. There are the major 4 types of protein structure. That is the primary structure, secondary structure, tertiary structure, and quaternary structure. Let's talk about the primary structure of the protein. Chemical structure of the polypeptide chain. The terms again, you need when I told polypeptide means, I think you know about what is a polypeptide, right? So don't confuse it. If you are still in confusion, ask to me. I can explain one more time. Chemical structure of the polypeptide chain in each protein, that is a number or sequence of amino acid residues linked by the peptide bond. So if you see the primary structure of the protein, it is just a sequence of amino acids. It is primary structure of the protein, nothing but a number or sequence of amino acid residues linked by the peptide bond. Means that each amino acid will be linked to another amino acid using peptide bond. This link will continue up to convert into the secondary structure, then secondary structure to tertiary structure, then tertiary structure to the quaternary structure. Then at the end, we will get a complete protein structure. That's why I told initially we will try to see the peptide, then polypeptide, then protein. So primary structure is the very, very important known thing to form a peptide. So let's say here only name itself, the number or sequence of the amino acid residues linked or together by the peptide bond. Let's say 2 amino acids are linked by the peptide bond. That is called peptide. If the linkage will be extended to up to 50, then peptide only. If the linkage will be extended to more than 50, then we are calling as a polypeptide. Then if it is linkage will be extended to the more, then protein size will be very, very big. So based on the number of and the sequence of amino acid only, we are determining the structure. I hope you understand what is the primary structure of the protein. If in the GPAD, if they will ask what is the primary structure of the protein, a number or sequence of the amino acid, or some other options will come, then you need to choose only sequence of amino acid. That is called only primary structure. When we are calling a primary structure of the protein means, if you see the simple amino acid sequence, then we are calling as a primary. I will see the complete structures in the next slide. Just remember, primary structure is a simple amino acid sequence linked by the peptide bond. If when we are seeing the sequence means 1 plus 1, 1 plus 2, 3 plus 1 plus 2 plus 3 plus 4 plus 5. That sequence can be linked together by the peptide bonds. That's about the primary structure. Again, one more competitive point. Sanger's reagents 1 fluoro 2 comma 2 4 dinitrobenzene. I will put this side. Yeah, Sanger's reagents and advanced reagents used for determination of the amino acid sequence. Amino acid sequence of the protein. If let's say then we have identified the primary sequence of amino acid by doing a synthesis or by joining amino acid by using a different bioinformatics softwares. When we joined amino acid 1 to 1, we need to determine either it is a primary structure or not, because simply joining is not valid, right? Whenever we are trying to do some experiments, whenever we are doing some research, we need to validate, we need to determine the whatever we have done. So let's say whenever we are trying to determine the primary amino acid sequence of the protein, that is called primary structure of the protein. So we need to use two reagents to validate the primary sequence of the we need to validate the primary structure of the protein or amino acid sequence of the protein. So these are the two reagents, very very important, the competitive point or the laboratory point. Sanger's reagents, you can see 1 fluoro, 1 2 3 4 5 6, 1 fluoro, 2 comma 4 dinitro. There is a two nitro groups are there. This is a benzene. So 1 fluoro 2 comma 4 dinitrobenzene also is a chemical name. Official reagent name is Sanger's reagents. Insulin protein can be determined by the Sanger's reagents. Insulin protein have 51 amino acids. You can note down. It's a very very important to the GPAT. I think they made me, I think last since for the more than 4 years back, I think they asked how many amino acid contain insulin protein. Answer is A, 51. A or B, C, D, but answer is 51. So Sanger's reagent will be used to the determination of the amino acid sequence, also used for the determination of the insulin protein. I think you know what is the importance of the insulin. Insulin is the kind of key hormone will be playing an important role in the diabetes mellitus. So there is a different kinds of insulins, insulin class, insulin pro, insulin lispro. There are different kinds of short acting insulins, long acting insulins. There are the different kinds of insulin. I think you know about it. Just I'm reminding here. Apart from the Sanger's reagent, one more reagent is that advanced reagent. This also called as phenyl isothiocyanate. These two reagents will be useful for the primary structure of the protein. Here, if you see in this slide, there is a two different kind of competitive bits. One competitive bit is that how we can identify the primary structure of the protein. Simple. Sequence of the amino acids residues. That is a simple way to do answer. And what are the reagents, what are the chemicals which are used to determine the amino acid sequence? That is Sanger's reagents and advanced reagents. So this is a very very important to the GPAT. I am not discussing broad detailed here. I don't want to confuse to you people. Only I am covering to the GPAT only. Yeah, sorry, the cursor is coming at the top of my slides. So I hope you understand what is the primary structure of the protein. Primary structure of the protein is nothing but a simple number or sequence of amino acid residues linked by the peptide bond. Two reagents are used to determine the primary sequence of the amino acid, also called as primary sequence of protein. So that primary sequence of protein is called primary structure. So primary structure of the protein is called a simple number or sequence of amino acid residues linked by the peptide bond. So these are the two reagents. I have explained 2 to 3 times. So if any doubts, you can ask at the end of the session. I'm going to move next slide. So yeah, secondary structure. I hope you understand what is the primary structure of the protein. Let's move to the secondary structure of the protein. Secondary structure of the protein is where we can see the special arrangement of the atom. When we want to see the special arrangement, we need to have a structure in the 3D. Three-dimensional is different, two-dimensional is a different. When we are writing a chemical structure in the blackboard, that is a 2D. The 2D, we can't able to see the interaction, we can't able to see the molecular coordinates. We can't able to see more number of features in a structure when we are drawing or in a blackboard or in a notes by using your pen. So if you see the 3D structure, we can't able to draw in a blackboard or in a note. For that, we need to have a proper software. In that software, we can see the three-dimensional structure of the molecule or three-dimensional structure of the protein. So in that kind of view of the special arrangement or the special arrangement of the protein can be seen in a secondary structure. In a primary structure, we cannot. Because primary structure is nothing but the sequence of amino acid, right? So in the secondary structure, we can see the special arrangement of the atoms or the molecules. The special arrangement of the protein by twisting the polypeptide chains. When there is a primary structure of the protein, there is a bioinformatics terms twisting or the looser segments or the domains or the subunits. I'm not going into the depth. Just remember that with the help of this primary structure, with the help of different kinds of term, different kinds of term, different types of term. There is a elysis, helix, sheets, twisting, the bricks. There are the different terms. Just no need to remember those terms. Just twisting or the bending are the different types of interaction between the polypeptide chains will form secondary structure. How primary structure will convert into secondary structure? Because of the foldings. Foldings, I think there are the different types of foldings. Again, I don't want to discuss everything here. So you just understand that after forming a primary structure of the protein, with the help of twist in between the organic reactions, in between the folding between one sequence of amino acid to another sequence of amino acid within the primary structure will form secondary structure. So the secondary structure nothing but any type of twisting or any types of foldings which is brought about by the linking or linkage of carbonyl and amide groups of backbones together by the means of hydrogen bond. Means that what I'm trying to say, if you see, this is the complete protein structure. If you see, this is I think I hope you know what is the basic skeleton of basic skeleton of the amino acid. This is one kind of amino acid. Let's talk about, this is a simple carbon amide group, a nitrogen, then this is a kind of R group attached to the another. This is the simple primary sequence of amino acid, or you can say simple primary structure. This is a one kind of primary sequence, this is another kind of primary sequence. There is a kind of dots are there, this is that bonds are forming, means that this is the kind of folding. Let's say this is the best example of loop. I think I know you people might heard about the term looping the things. So this is a kind of loop, a kind of folding. You can see the folding, how the amino acids structure is the folding. Each fold, there might be a one either peptide bond or hydrogen bond is important. Peptide bond will be definitely easier, but you can see hydrogen bond is also trying to form here. So the primary sequence of the sequence of amino acids or the primary structure of the protein will convert into the foldings, will convert into the twist, then it will form secondary structure. So let's say if you see, if you can able to see in my slide, use the cursor. Yeah, I got the cursor. So valine, histidine, tyrosine, like that. In this way, sequence will form. This sequence will convert into the folds. Like that, loop will continue. This loop we are calling as a alpha helix. I hope you understand. Valine, histidine, tyrosine. These are the amino acids. Amino acids are linking. In a biological spectrum, is the different to understand the things. That's why I converted into the pictorial representation. If you open human body, you cannot able to find any protein. Everything will be blood, organs will be there. each cell have a kind of there is a neurons signaling pathway between one cell to another cell. In between only receptors, in between proteins, enzymes will be there. So it's a very tough job to getting a live. That's why always biology is there to understand these things. So missing the cursor, sorry for inconvenience. So valine, yeah, I got cursor. So valine is the one amino acid linked to the histidine. Histidine is an amino acid linked to the tyrosine. In this way, linkage or the sequence will form. This is called a primary structure. This primary structure will convert into the folds. It will convert into the such type of folds or the twisting between the amino acid to another amino acid. This loop will form. This loop we are calling as a alpha helix. After doing this, after creating this loop, then sheet will form. This we are calling as a beta palleted sheet. So in the GPAD, if they will ask which of the portions or the which of the part or the which of the segments can be identified in the secondary of the secondary protein structure. Option A, alpha helix and beta plated sheet. These options are very correct. Remaining options can be anything. So if I will give all 4 types of options, time will waste. That's why I'm giving only correct option. So option will be alpha helix and beta sheet. So how we can identify the secondary structure with the help of alpha helix and beta sheets. So these are the 2 ways asking questions. So how to identify the protein secondary structure of the protein is alpha helix and a beta plated sheets. You know how we can get the alpha helix and how we can get a beta sheet. Primary sequence of amino acid or the primary structure will convert into the folds, then it will form helix. Upon certain time, then it will convert into the beta sheet. So that is the clear-cut idea about the secondary structure of the protein. So basically, in the secondary structure, we need to remember the alpha helix and the beta sheet, how it will form with the help of foldings and with the help of fist. So again, there might be a different bonds, although there here bonds are not important. In one slide, I have explaining the all types of the bonds which are involved in the protein structure. So basically, there might be one more important question to the GPAD, where we can able to observe the hydrogen bond formation. Only hydrogen bond formation, not complete hydrogen bond. Hydrogen bond formation can be seen by the secondary structure of the protein. Please note on this point. Hydrogen bond formation can be seen by secondary structure, only formation. When the amino acid sequence, when the protein structure, while doing the loop or while doing the foldings, there might be a form of hydrogen bond between one fold to another fold. You can see here dotted. See, hydrogen bond is forming between hydrogen to the oxygen. In between, a peptide bond will be also there. Peptide bond will be used to bond between one amino acid to another amino acid. Hydrogen bond will be used to form between one sheet to another helix or one fold to another fold. So that's the 2 kind of important things in this slide. Please note down it. Again, I am repeating. Alpha helix and beta sheets will be used to form the secondary structure of the protein. Hydrogen bond is to form between one helix to one sheet, or one helix to another helix, or one helix to another sheet. So with the help of hydrogen bond, these formation will be happen. In between amino acid to another amino acid, again peptide bond will form. So that's the brief intro about the secondary structure. So there is important again. Ramachandran is an Indian scientist. Is determined are the invention these kind of sheets and helix. Ramachandran is the scientist. Is an Indian scientist. Is developed these a plot is called Ramachandran plot. This plot used to determine the production or determine the secondary structure of the protein. What is the primary structure of the protein? How we can determine? With the help of 2 reagents. What is the Sanger's reagent and Adman reagents? How we can determine or predict the secondary structure of the protein? When we are trying to do a synthesis of the protein, when we are trying to make a protein, when we are trying to develop a protein, we need to validate. Every stage validation is required. In the stage 1, that is called primary structure, we are using 2 reagents. In a secondary structure of the protein, we are using a Ramachandran plot. So now you need to understand in a GPAD, if they will ask like that, how we can which type of the plots can be used to determine the secondary structure of the protein. There is a different plots, but one important plot everyone is using, even I use it in my project. that plot name is called a Ramachandran plot. There is a Arad plot, a red plot. There is a different plots are there, but famous plot. Most of the things can be easy to determine the secondary structure is that Ramachandran plot. Let's talk about the Ramachandran plot. What is it and how we can determine it? So this is the Ramachandran plot. He is introduced this kind of plot when he is doing his research. So Ramachandran plot have a 4 coordinates: coordinate 1, coordinate 2, coordinate 3, coordinate 4. These coordinates he has a scaled. The scale is 0 to 180 angle. We can say angle or decrease. With the help of 2 terms, that is called psi angle, pi angle. By using these 2 angles, he described this plot between x-axis to y-axis. Y-axis, he has given the y-axis is denoted by the pi angle. I will give the symbols in next slides. X-axis can be denoted or the identified by the psi angle. So let's say in x-axis, you can see minus 180 to 0 is called a minus pi. A 0 to plus 180 is called plus pi. This is x-axis scale. In x-axis, x-axis denoted by the pi angle. Pi angle, you can see plus 180 to minus 180, 0 to minus 180, 0 to minus 180. In the GPAD prospect, I think Ramachandran plot will be very, very important to the NIPER. But again, we can learn for GPAD also. No, nothing will be wrong here. The exam point of view, they will ask where we can see a right-handed alpha helix, where we can see left-handed alpha helix, where we can see beta sheet. So when they will ask in the such a question, we need to identify minus 0 to minus 180 of minus 5 to minus 180 of 0 minus psi minus pi. This coordinate will be giving the right-handed alpha helix. So this is the third coordinate. Third coordinate of Ramachandran plot will be used to determine the right-handed alpha. It is a very going into the depth. It is a very, very important for the GATE exam life sciences. I don't think so they will ask this much depth in the GPAD, but try to learn, try to understand the things. Anyway, the NIPER prospect I will teach one more time, complete about this Ramachandran plot and softwares, about what are the softwares which are used in the bioinformatics. So these are the 4 coordinates. Each coordinate will give a kind of spectrum to identify the secondary structure of the protein. The secondary structure mainly deals with the helix and alpha helix and beta sheets. So this is the beta sheet. Again, alpha helix will be 2 types: left-handed alpha helix and right-handed alpha helix. So right-handed alpha helix will be involved in the third coordinate. Left-handed helix will be involved in the second coordinate. So to identify if you in the plot after preparing a secondary structure of the protein by using the software, if you determine the Ramachandran plot, this alpha helix we should get this region. The region name as 0 to minus 180. Again here also 0 to minus 180 of psi and pi. Then only we are able to determine the secondary structure. We can assume that we can predict that we got the exact 100% purity of the secondary structure. Okay, I hope you understand how to determine the secondary structure by using the Ramachandran. So how to identify the beta sheet of the secondary structure? Again, only plus psi means that 0 to 180 degrees. So I think they won't ask how to determine. They will ask what is the Ramachandran plot x-axis. You can say plus pi. You can say basically pi. Again, pi will be different into plus phi minus phi, pi, pi. Then psi. This is a y-axis. Y-axis can be plus psi and minus psi. So that is a clear-cut idea about the axis in the Ramachandran plot and the coordinates in the Ramachandran plot. Basically 4 coordinates. A 4th coordinate will be nothing. If there is any overlap, this sheet and alpha helix here, then our prediction is going to wrong. It should be this angle only. This should be this angle only. This should be this angle only. So it is a advanced level of the discussion. Just my understanding I am giving to you, but don't worry if you understand it and try to understand it. If not, don't worry about it. After qualifying the GPAT, we will discuss in the NIPER. Again, protein protein have the 4 different protein structure. Protein have the 4 different structure: primary, secondary, tertiary, quaternary. Within the secondary, there are the different types of are there. That is that already I explained: helix and sheets. So there are the three common secondary structures in the protein. When we identify the helix, when we identify the sheets, when we identify the turns, then we can think, or we can assume that this is the protein, this is the secondary structure of the protein. So there are the three common secondary structure in the protein, namely alpha helices, beta sheets, and turns. So these are the three key fragments, or the agents, or the structures which we can observe in the secondary structure of the protein. So if they will ask in the GPAT, what are the different things which we can identify in the secondary structure of the protein? Alpha helices, beta sheets, and turn. That is a one way of asking question. Second way, how we can identify the secondary structure of the protein? Option A, with the help of alpha helices, beta sheets, and turns. So basically, we used to identify the alpha helices and beta sheets, and turns are also a very, very important segment of the parts in the secondary structure of the protein. I hope you understand well known about the secondary structure of the protein, how to determine the protein secondary structure of the protein, what are the different types of the secondary structures. So I hope with this discussion, I'm going to the next slide.